Welcome to Dr. Balaga's God Knowledge Doc podcast, your quick dive into the latest medical breakthroughs to keep you ahead of the curve. Let's get started. Today is October 22nd, 2025. This video is on a Nature Communications article titled Sex Stratified Genome Wide Association Meta Analysis of Major Depressive Disorder. It's accompanied by a News and Views article in Nature today titled Sex Differences in Depression Revealed by a Large Genetic Study. It was published online today. Major depressive disorder affects twice as many women as men worldwide. Women more often present with atypical and immunometabolic depression, including weight gain and hypersomnia. Men with anger, risk-taking, and substance abuse. Biological factors include the HPA, hypothalamo-pituitary axis, and the HPG axis dysregulation, immune activation, and sex hormones. The hypothesis is sex-specific genetic architecture partly explains these differences. Methods utilized: sex-stratified GWAS plus genotype by sex meta-analysis across 5 international cohorts with previous PGC data. 130,471 cases and 159,521 controls were enrolled. Females were enrolled. 64,805 male cases were enrolled and 132,185 control males were enrolled. Diagnostic criteria based on diagnostics and statistical manual of mental disorders. Chromosomal sex was used, XX versus XY. Statistical tools, including the MLM model, the meta-analysis, heritability bias Bayes S, and LDSC, polygenic overlap, replication, generation Scotland, about 9,000 individuals. Lifetime prevalence assumed 0.2 in females versus 0.1 in males for genome-wide significance. Results: there were 16 genome-wide significant SNPs in females and 8 in males. There was including a novel X chromosome, IL-1 RAPL1. Heritability was high in females, 11.3%, compared to 9.2% in men, p-value 0.001. Greater polygenicity in women, 13,000 causal variants versus only 7,000 in men. There was substantial but not complete genetic overlap. Major depressive disorders showed a strong genetic correlation with body mass index and metabolic syndrome. Only one shared gene, NEGR1. Also, there were female-specific neuronal development gene sets enriched. Key limitations of the study: predominantly European ancestry, female sample 1.6 times larger, possible male underdiagnosis, and sex-aware quality control constraints. Key takeaways: females carry a higher genetic burden for major depressive disorder risk. Males show unique X-chromosomal signals. Sex-specific pleiotropy links female major depressive disorder to metabolic traits. Precision psychiatry must incorporate sex-specific genomic risk. Clinical pulse: depression is biologically dimorphic. Clinical phenotype mirrors genetic architecture. Female patients with metabolic syndrome or obesity may represent immunometabolic subtype. Consider sex-specific screening for metabolic risk in major depressive disorders. Pharmacogenomic therapies and biomarker trials should use gender-stratified designs. Integration of multiomics and hormonal profiles could enhance therapeutic response prediction. Eponyms Blockland et al. Were first stratified major depressive disorder GWAS foundation for in 2020 and was the foundation for current study. The IL-1 RAPL1 is the interleukin-1 receptor accessory protein like 1, is linked to neurodevelopment and memory. The major depressive disorder polygenicity was pioneered by Brendan Bullock Sullivan et al. This study underscores her genes, his genes, and our minds, a new frontier in gender-aware genomics. The accompanying news and views article discussed this paper. They said women have nearly twice the risk of major depressive disorder as men, a platform transcending cultures and diagnostic practices. Using a sex-stratified genome-wide study of over 200,000 individuals, Thomas et al. Uncovered 13,200 causal variants in women and 7,100 in men, revealing 6,100 female-specific variants. The key findings: female-specific genetic effects, 3 genomic regions linked to major depressive disorder in women only, metabolic links stronger in women, major depressive disorder in women showed tighter genetic correlations with body mass index and metabolic syndrome, male-specific signal, 1 novel X-chromosome variant, IL-1, RAPL1, associated with major depressive disorder in men, a gene tied to memory and neurodevelopment. Overall genetic correlation, about R0.9, suggests large but incomplete overlap between sexes. The implications of the study: women bear a heavier polygenic load for depression risk. Findings underscore the need for sex-aware psychiatric genetics and precision therapies tailored to biological sex. Future research should integrate molecular intermediates, gene expression, and hormones to clarify how genetic risk translates to disease expression. If you enjoyed this episode, share it with your colleagues. When we change how we see disease, we change how we shape lives. Until next time, stay curious, stay compassionate, and keep seeking wisdom. Podcast brought to you by masterbedfacts.com. This podcast is for physicians and healthcare providers. If you enjoyed this video, please like, subscribe, and forward to your friends.